Everybody, good afternoon, and we appreciate your time joining us for this webinar. I'm Brian Guess. I'm a technical sales representative for LALNIX, and I'm going to be hosting this webinar today. We packed a lot of good information about our nematicide LALNIX ACT DC. And so the title of our presentation is Protecting Your Crops from Plant-Parasitic Nematodes with our nematicide LALNIX ACT DC, which was formerly named MeloCon LC. Our agenda for today, we're going to briefly touch on who is LALNIX. We're going to do a deep dive into the product, LALNIX ACT DC. Dr. Megan Rains is going to be reviewing trial results, best use practices. We're going to really spend some time on LALNIX's rehydration protocol and what we've learned to really maximize the efficacy of this product. We'll review the key takeaways, and then at the very end, questions and answers. Okay, I introduced myself. I'm the technical sales representative here supporting the sales side. Dr. Megan Rains, she's had over 10 years of experience in chemical, biochemical, and microbial research and development. She graduated from Queen's University with a PhD in biology, where she studied the chemical composition and molecular regulation of lipid barriers. She also possesses a master's and bachelor degree in chemistry from the University of Toronto and the University of Western Ontario. Within LALNIX, Megan runs the Plant Care Field Solutions team. She and her team are responsible for the technical support of the LALNIX products in our portfolio. Megan oversees product efficacy trials, compatibility tests, and a multitude of other responsibilities that support the commercial sales of our products. And with a little introduction, I'm going to go and get started here. Okay, a little bit about LALNIX. We are a privately held company established over 100 years ago by Fred Ed LALNIX. Fred was a German immigrant, migrated to North America during the 19th century, and he started selling yeast to bakers, and that's how LALNIX was born. Over the years, we've expanded and grown as a company into other market segments, but we've really still stayed laser focused on the research and development, commercialization, and manufacturing of microbial-based products. These would include your yeast, fungi, and bacteria products. We have 12 business units globally. Some of the larger business units would be your animal nutrition, our baking division, and our enology. Dr. Megan Rains and myself, we're part of the plant care division supporting the sales into North America. Next slide. Here is our global footprint. We are a globally based company. You can see the red dots are our manufacturing plants. Our turquoise dots, those are our research facilities. We have some in North America, over in Europe. And then the sales representatives are the yellow dots, and you can see we're strategically located throughout North America and the US, supporting our specialty crop and our row crop divisions. And then we sell globally, East Asia, Australia, South America, and Europe. And moving on to our product portfolio, we're going to go ahead and start on the top right-hand side of the slide, over under the fungicide category. These are the products that we have available and commercially available to sell into the United States. The fungicides would be Wallstop G46 and Wallstop K61. Moving down to our insecticides, we have two different formulations of the same product, LALGuard M52 OD and LALGuard M52 Granular. The granular is certified organic. Moving down to the bottom right-hand corner, our nematicide, which we'll be talking about today, is LALNIX ACT DC. Moving to the left, bottom left of the screen, our stress reducer, LALSTIM Osmo. Biofertilizer would be LALRISE Vita, and then the top left hand of the slide, our mycorrhizal inoculant, which is LALRISE Max. All these products are commercially available in the United States, and I think all of them are certified organic except our LALGuard M52 OD. Due to the formulation, we are not able to receive organic certification on that insecticide. And then moving on. So today's product, LALNIX ACT DC, it's an OMRI-listed bionematicide. It's applied in the root zone, targeting those plant-parasitic nematodes. The active ingredient is Perpetuacillium strain 251. And this is a premium Perpetuacillium formulation with the backbone of strain 251 that is parasitizing those nematodes. We see parasitism in the nematode eggs, juveniles, and adult females. The formulation is a dispersible concentrate, hence the DC in the name. We have a shelf life of 2 years at 68 degrees. And that's a key. If you want to have achieved a 2-year shelf life of this product, 68 degrees is that required temperature. We have some other storage temperatures that we're going to review later in this presentation for if you're going to be using it this season, or if you need to carry some over to the next season, you'll need to keep it at that 68 degrees. We have our package sizing of 34 fluid ounces per bottle and a recommended application rate of 10.25 fluid ounces per acre. Very simplified application rate on our label. It's really 10.25 fluid ounces is what we have on the label. And then moving on, I'm going to pass it off to Dr. Megan Rains, who's going to dig into some more of the science and history of the presentation. Thank you.
So thank you, Brian, for that lovely introduction. As Brian mentioned, my name is Megan Rains. I'm based in Sault Ste. Marie, Ontario, Canada. I spend a lot of time on the West Coast, and Brian and I were actually just in the desert where it was a balmy 110. So I'm happy to be back in the cold for the time being. Yeah, so I just wanted to take some time to kind of introduce the product and its history to you. And as you can see from the slide, our Purple Psyllium product was developed not kind of overnight. It's been a long history. And it was first identified in 1910, where it was actually classified as a penicillium. It was later reclassified into a purpacillium, but we did see that first association with nematode eggs as early as 1966. So this is a strain, and this is a species that has long been known to act on nematodes, specifically on the eggs, juveniles, and females. So starting in the 80s, we saw kind of a period of commercialization. And then probably the most significant portion of this for our history is in 2003, the strain and the data rights were acquired by a company in Germany called Profita. And what they did is they began a period of about 10 to 12 years where they developed these formulations based on these Purpacillium strains, Purpacillium lilacanum 251, where they developed the WG, WP, and DC formulations. And this was eventually purchased by Bayer and marketed under the brand name of BioAct. So until 2021, when Lallemand actually acquired the Profita facility, which was a Bayer facility at the time, where we took over not only the production, but we actually took over the manufacturing facility so that we could supply the market with these robustly studied products. So it was distributed until this year under the MeloCon brand name by Certis, but in 2024, Lallemand took back the distribution, where we continue to sort of develop our field and lab programs and continue to bring value to the market. So that's kind of a big part of what Lallemand strives to do, is we strive to bring value to our growers. So in order to do that, we need to understand what the problem is that we're hoping to address. And I'm sure it's no surprise to anyone on this call that we're going to be talking about nematodes today. And nematodes are actually probably the most abundant multicellular organisms on Earth. And there's about 20,000 different described species of nematodes. And fortunately for us, not all of those are a problem, and most of these are actually beneficial to the soils in which they dwell. So they'll do things like feed on bacteria, supply organic matter, and kind of live in a symbiosis with other soil microorganisms. But unfortunately, there are a class of nematodes known as plant-parasitic nematodes, where they contribute to somewhere around $80 billion of damages worldwide. So there are some strategies that growers have traditionally used to control the nematode population. Among those are farm hygiene, so not transporting materials that could be infected with nematodes from one location to a secondary location where we don't have a previous nematode population. Monitoring your fields for signs and symptoms, crop rotation, even going as far as leaving your ground fallow if necessary to reduce those populations. Soil management, especially in those sandier soils where nematode populations tend to be higher. Work on things like building organic matter. You can also use nematode-resistant crops, solarize your soil kind of in those warmer months, and then have targeted applications of your nematicides when the nematodes are going to be most active, so those warmer summer months. There's a variety of different chemical and biological controls, chemical nematicides, so these are your vidates, your celebros, your vellums of the world, as well as biological nematicides, which is what we're going to spend some time focusing on today, like LALNIX ACT DC, which is a bionematicide. So LALNIX ACT DC can actually control a much larger range of nematodes than we have listed here, but these are sort of the, I think I put 10 most common or 8 most common, 8 I did. I think there's 18 on our label. So if your particular nematode species that you're looking to control is not present here, the label will have a comprehensive list of everything in which we are registered to control. Probably the most significant species on this list is going to be your root knot nematode. So these tend to be the ones we come across the most often, as well as cyst nematodes. And you can see here on the right just a photo of the nematode eggs being attacked by the Purpocillium contained within LALNIX ACT DC, and you see the sort of 4 eggs that are, I'd say, they've been parasitized, they're flaccid, and then you see the fungi actually moving in to, attack the rest of the eggs. So we have good efficacy on a wide range of species, including probably the 8 most common that we deal with. So the active ingredient, as Brian had mentioned, is spores of the Purpocillium lilacinum 251 fungi. And the way it works is that these spores must contact the nematode or the nematode eggs. So as those nematodes move through the soil, they will come into contact with the spores of LALNIX ACT DC. And when that happens, you'll actually have the spores germinate and produce mycelia. So once you see those, or once, I guess, the second photo here, you can see the spores germinating. You'll actually get hyphae that will engulf the host, building a prosoria and penetrate the eggs and juveniles and adult females with physical pressure as well as enzymes that help to break down kind of the exoskeleton and the egg mass. And then once it actually breaks into either the egg or the juvenile or the adult female, the fungus will kill the nematode by feeding on its body contents. So sounds pretty cool, if you ask me. But really the thing we need to be aware of is that we have to have the fungi get into contact with the nematode or the nematode eggs. So when we're applying this product, the objective is always going to be to get the spores into the plant root system, since that's where the nematodes are going to be living. There's a few different ways in which we can do this. Probably the most effective way to get the spores onto the nematodes or the eggs is through drip irrigation or another soil-directed irrigation system. If we don't have access to drip irrigation, you can also use a soil-directed spray around the plants or along the rows of the soil surface. And if you use a soil-directed spray, you're going to want to drench the spores in the product with irrigation water with something like overhead irrigation, as long as you have an open canopy, to really drive those spores down. And if we can't do that, the last choice is really a mechanical incorporation. So this is kind of something we would recommend that you do before planting, so you can really drive it in. And this can be done, we usually recommend like, I think 7 to 14 days before planting if possible. But as close to the day before is also very efficacious. And then we tend to see mechanical incorporation in row crops where we don't have irrigation systems in place. So things like potato, carrots, for example. But what we want to remember is that water is going to be the best transport medium in order to move the fungal spores into the root zone, and that the amount of water can always vary depending on your soil type. So if we have a clay soil versus a sandy loam, you might have to adjust your water volumes, but we want to make sure we get those spores to the root system. So as Brian mentioned, it is a water-free formulation. So we want to actually look at what happens when we dissolve this in water. So when we actually put LALNIX ACT DC into water, as you can see on the left here, we have almost an immediate dispersion through the water. So it's highly water soluble. We also ran this kind of with a competitor product off to the side here. Competitor product is at a field rate of 2 pounds per acre. It's a WP formulation, whereas the DC formulation of LALNIX ACT DC had much better dispersibility in water and a homogeneous solution formed within 30 seconds. As we speed up the video to let our competitor kind of catch up and catch up some more, we see that we still have that floating powder. And with that floating powder, it actually did not form a homogeneous solution even after 7 minutes. So from the top, you can see sort of the clumping of the product that hasn't quite gone into solution with 72 degree water, as well as kind of that settling out at the bottom. And what this really means for us is that when we look at our filter systems, we tend to see kind of that settling out and it getting stuck to the screens, which means we're going to have to clean our filters a little bit more often when we consider some of these powder products. So as I mentioned, water is critical to ensure we get the product delivered where it needs to go. So we want to recommend that if your final concentration after dilution and kind of your final volume is going to be less than 0.075%, and even ideally 0.05%, you don't need additional water in order to drench it in. The concentration of the spores within the water is sufficiently low that there's enough water to kind of move the spores into the root zone. But if your final concentration is going to be greater than the 0.075%, we always recommend using some irrigation water to really drive the spores into the root zone. And the reason for this is that if the spores are too concentrated on the topsoil, then you're going to get an overdosing at the wrong place. So nematodes aren't just laying the eggs at kind of in the roots of right at the crown. You're going to have that entire root ball being affected by nematodes. And as a result, we need to make sure that we get the product drenched down into the root system. So when we actually look at where the spores go when we put the product on plants through drippers, what we saw is that about 70 to 90% of all of the recovered spores are found within a 4-inch depth and an 8-inch diameter. And if we look a little bit beyond that, within about 14 inches in diameter beyond the center, then we can kind of classify, we catch about 5 to 20%. Again, this is going to be very soil type specific. And if we go even further down between the 4 to 8 inches below that central zone, we get a further 3 to 11% of the spores. And deeper than 8 inches, only 1% of spores are found. So this is really just to drive home the point that we need to make sure we are putting sufficient water with the product in order to get the spores where they need to go and in order to treat the entire root mass of the plant. So the best way to do this is making sure that your final concentration when you're applying it is kind of below that 0.075%, ideally around 0.05%. The other thing to note about LALNIX ACT DC is that it is sufficiently temperature tolerant. If we look at the growth on Petri dishes at constant temperatures, so we can see 0, 10 days, and 15 days at a temperature range of 75 to 97. What we can see is that, the higher the temperature, the lower the radial growth on a Petri dish. But what it means is that we have an effective range and we have an optimum range. So the effective range is going to be where the Purpacillium is capable of growing, and the optimum temperature is where it's going to be where it grows best. So the labs have determined that the effective range is between 64 and 86 Fahrenheit with an optimum temperature of 77 Fahrenheit. And we know that we grow in a variety of climates and that, optimum temperatures are not always the case. But good news is that the Purpacillium can actually withstand soil temperatures over the season in hot production areas because established fungi can endure very severe conditions for some time. Now, that's not to say they can survive indefinitely at 100, but they can survive short bits and certainly long enough to get the irrigation water flowing and those soil temperatures kind of coming down. So LALNIX ACT DC, although it has efficacy on eggs, juveniles, and adult females, which we know the adult females are the real trouble causers, it is going to be most active on eggs as well as those early juvenile stages. So as in this case, the root knot nematode or Melodagine moves through its life cycle from an egg shown here on the kind of upper left and moves through its various juvenile stages, and we see it moving into the root structure and through the cortex, and then eventually, germinating and producing galls. We have LALNIX ACT being most active on the egg structures, but also having activity on the juveniles. So if we look at the very nice electron microscope images here, we actually see the fungi colonizing on the eggs as well as juveniles. So this is a J2 juvenile. And we can actually visualize this in the galls where an untreated root structure shows an incredible amount of staining, which is indicative of nematode galls, versus a LALNIX ACT DC treated root structure where we don't see that level of blue color and staining. And it's not to say it's going to completely eradicate the population. We can still see some staining of the roots, and you can see some of those gall-like structures, but we do have a significant reduction. So as I mentioned in the history bit about this, we have a vast amount of data that's been accumulated over many years, and it's been about 45 years of research that's gone into the development of these products. And what that's led to is a significant number of field trials that have also been done, and it's a very highly documented product through a variety of GEP and extrapolation trials. And if you're interested, those codes and brackets are the EPPO codes. So just kind of some international nomenclature there. But it's just a widely studied product. And to get through some of the trial data, we actually can see that treatment with LALNIX ACT DC, whether it be in a standalone program or an IPM program where it's kind of combined with conventional nematicides, we see a significant reduction in the number of eggs. And in this case, we're looking at cucumber and tomato. We can also see that translate into the yield on tomato. So in this case, it's against oxymel, which is the active ingredient of Vidate. So untreated on the far left, LALNIX ACT DC with 4 applications in the center in the red, and then the chemical control showing equivalent or very similar yields. So we see less eggs leading to number of fruits being increased or a higher yield. And this trial was done in Tallahassee, Alabama. We see a similar result in carrots. So if we look at juveniles in this case, as well as eggs, we see significant reductions. So this was done in microplots. And with the significant reduction, it leads to less galling in the carrots and obviously higher yields. So they did this in both a field study as well as a greenhouse study, finding the same thing. And as mentioned, there's been quite a few studies done, and we have, a very large repertoire of data for cucurbits, solanaceous crops, lettuce, carrots, sweet potato, things like that, where we see a consistent improvement of yield and a reduction of eggs and juveniles, but ultimately we care about yield when compared to not treating your crops at all. We can also look at this relative to chemical controls, and we usually find we're about on par or slightly better, slightly worse, just depending on the trial conditions, but we're always kind of in the same realm as a lot of these chemical products, and definitely better than not treating at all. If we look over kind of the course of all of what we've done for standalone programs, we can see yield increases in 88% of all of our trials when looking at tomatoes, and very similarly, 92% increase in all the trials when we look at cucumbers. The other type of trial we like to run is IPM type trials. So this is some sweet potato data that's a little bit more recent, so done in North Carolina. And we can see on the left here, the non-treated control, oxymel, so Vidate, as well as LALNIX ACT DC applied twice, or we have a combined program where oxymel was applied first, followed by LALNIX. And we can see that the Vidate, the LALNIX ACT DC applied twice, as well as the oxymel, then LALNIX, all had significant improvements to the yield. So in this case, it was done in pounds of sweet potato per plot that it was harvested on. And then on the right, in a similar study done the following year in Raleigh, North Carolina, we see similar results where the two applications of LALNIX ACT DC functions as well as the combination of the LALNIX applied with oxymel, or Vidate in this case, improving the yield significantly over the non-treated controls. So we can see sort of a seamless integration into sort of your conventional chemical approaches for nematode control. We also have an established compatibility database, so we can also use a variety of tank mixes, or you can apply these rotationally, or use LALNIX ACT DC to really drive down the amount of chemicals that you're putting out. These are all great options. So we did see that there was no adverse effects on the survival of entomopathogenic nematodes, so your soil beneficial nematodes. We also saw no effect on mycorrhization and no effect on the growth rates of other biocontrol fungi, which is great news. So it's really just attacking the nematodes and leaving all the beneficials alone. So in order to really use this product correctly, what we want to touch on today is really this rehydration protocol. So before you use it, what we'd like you to do is to gently rehydrate the spores. And I've mentioned that this is a water-free formulation, which means that, we have to introduce small amounts of water back in order to maximize the efficacy. And we'll go through kind of a step-by-step, and Brian made a really nice video that shows him carrying out this protocol to show you just how easy it is. But after you've rehydrated the spores with that water, whether it's on a small scale or a much larger scale, like a 5-gallon pail, then you're going to be mixing it into a full water volume, making sure we add our product last. We also want to highlight that you want to pay attention to PPE requirements and use agitation, but also avoid foam. So whether it's with an anti-foam or just with the sheer volume of water, there's kind of some steps we're going to walk you through here. So the first step of the rehydration protocol is really to allow the product to acclimate unopened. And the reason for this is that the spores within LALNIX ACT DC are a living microorganism. And if you think about how humans are in general and, our kind of tolerance to the cold, I think all of us are much happier when it's room temperature. And it's much the same for bacteria. They're going to function a lot better when they are at optimal sort of conditions. And we talked kind of about the Purpacillium being very temperature resilient, but it has an optimum temperature closer to that 77 Fahrenheit. So we want to allow the unopened product to kind of come to room temperature and warm up, especially if you're storing the product cold. So maximizing shelf life, we just want to be kind of very cognizant that we might need some time to let the product warm up before use and taking it out of cold storage. So ideally, 1 day prior to use, this can be as short as overnight. It's not a big deal, but give it some time to get a little bit warmer. For small scale applications, really what you want to do is use the lid as a measuring cup and add 2 capfuls of lukewarm water, seal it back up, shake it, and allow it to stand. So this is kind of product that's sat out overnight, put 2 capfuls of warm water in, and we're good. If you let it stand for a minute, it kind of is happy, and then we can dilute it and use it. For larger scale applications, so these are going to be kind of big pivots, or you just have a lot of acres you're treating that day, you can actually use something like a 5-gallon pail, which most farms have, and combine it with lukewarm water in a ratio between 10-to-1 LALNIX ACT DC to water to 14-to-1. And the reason for this is we don't want you to overfill the bucket, but you just kind of put the product in, put a little bit of warm water in, stir it up, and allow it to sit. And then you can continue with your mixing and loading procedures. So here we have Brian to demonstrate.
Hello, I'm Brian Guess with LALMON, and I'm going to be showing you the LALMON rehydration protocol for our nematicide LALNIX ACT DC. The formulation of this product is a water-free concentrate, so it's critical that we add a small amount of water back into the product before the application. By following the LALMON rehydration protocol, you will be maximizing the efficacy and performance of this nematicide. A critical component of this rehydration protocol is the temperature of water that you're using for the rehydration. It needs to be 63 degrees or warmer. It can either be from a well or from whatever your typical water source is for applying pesticides. I have my thermometer. I'm going to take a reading of the temperature, 72 degrees. So we exceed our 63 degrees or more temperature requirement. I'm going to put on the appropriate PPE. I will take 2 full capfuls of water and apply it into the master container of the product. 1 and 2 capfuls of water. We're going to close the lid tightly. And now we're going to shake the product vigorously to rehydrate. You will let it sit for 1 minute, and now you are ready to apply the product. You've completed the LALMON rehydration protocol. A call out is foaming. Foaming can be detriment to the efficacy of this nematicide, so we recommend using an anti-foaming product in conjunction with LALNIX ACT DC when you're applying the product. If you have any specific questions, please reach out to your LALMON representative. Thank you.
So once we have that gentle rehydration protocol that Brian expertly showed us how to use, and we saw how simple it is, then we can actually continue with mixing and loading. And proper mixing is going to be something that we want to take care with. So the idea here is that you are going to need to determine how much water you need and use the full water volume in the tank. So fill your tank with the full volume prior to adding the product. Once you have the water in the tank, you're going to turn on the agitation and add the required amount of LALNIX ACT DC to that tank. You can then triple rinse the product container and empty that into the tank as well. So as Brian mentioned, we want to avoid foam generation. So the way we do this is by avoiding air intake into the suspension. So keep your water pressure low at filling, don't use your induction hoppers, kind of all of the best use practices to avoiding foam, and turn off the agitation when the tank level gets too low. So if you need multiple tanks of product to go out and be diluted, turn off that agitation when the tank contents gets too low that you start to see foam being generated. Another piece of advice is to always add the product as the last step. So once the full water volume is in the tank and we have the agitation turned on, that's when you want to be adding your product. And if you would like to, or if you're still seeing foam, you can use an anti-foam prior to filling the tank with water. And then just some storage requirements. As mentioned, if you can store the product at 68 Fahrenheit, you get 2 years of shelf life, and up as high as 86 is 6 months shelf life. And we just want to reiterate, you want to avoid prolonged and repeated exposures to temperatures of 104 for longer than a week, and please avoid freezing the product. So with that, I'm going to hand it over to Brian to do kind of a label review and give some more good information and wrap it up.
Excellent. Dr. Rains, thank you for that. I always learn something new on your presentations, and the way you pronunciate the active ingredients and the scientific terms is very eloquent. Before I dive into the label, I wanted to go back and just talk through with you, Megan, or Dr. Rains, about the Oxymil or the Vitate program and the yield differences or the comparable yields with the LALNIX treatment. Selling a nematicide is so different than any other insecticide or fungicide. No one's really looking at the eggs or the nematodes in the soil. That's really, that yield is how you're evaluating it. So for the individuals that are on this call, either if you're a sales representative from a distributor or a grower, we'd love to do some side-to-side treatments in your fields or your customer fields, putting the LALNIX up against your synthetic program or your conventional grower standard. A lot of times I think there's a yield drag in fields, and we don't know, is it a disease? Is it a nematode? What's truly happening in there? And a lot of times it could potentially be nematode damage that is compounded by an infection or other issues in the field. So incorporating LALNIX in a trial competing against your grower standard, I think could be a way to visually sell that or to show its performance in the field. And this specific trial, if you're looking to reduce your overall toxicity, I think LALNIX, you go in with your pre-plant fumigation or your pre-plant treatment. You can replace your Oxymil or your Vitate treatment on the front end with a LALNIX, reducing your overall toxicity in your program, and then followed up with your second application would be your synthetic material for your non-organic growers. And Megan, how do you see, I wouldn't say selling the product, but introducing the product where you're not seeing the dead nematodes in the soil? It's such a challenge. Really, yield, I think, is the best matrix to measure it. And
yeah, yield is always going to be kind of your gold standard metric. It's one thing to come out and say, we parasitize eggs and we parasitize the juveniles, leading to death. But ultimately, if that doesn't end up in a yield response, then, it's, I won't say wasted input, but it's hard to quantify the actual role of that nematicide without a yield response. So I think yield should always be something that we're considering. And I think we can clearly see that both as a standalone treatment as well as an incorporation with kind of conventional products, we do see that yield response. And as an added benefit, it's significantly less toxic than using something like a Vitate. And it also helps to maintain the soil microbiome, where you're going to have a lot of those beneficials existing and helping with fertility and things like that. It's just kind of an added benefit that we can reduce the amount of chemical while still maintaining that yield response. And I think it's kind of something we always want to be evaluating and mindful of.
And yeah, this data excites you as much as it does us. Please reach out. We're going to have a map of the local Lawamon reps, and we'd love to come out, do a grower demo, or even just a side-by-side, one field treated and one field untreated, and really show you firsthand the performance of this product. with that, I'm going to go ahead and dig into the label. So we have two labels in the marketplace for LALNIX ACT DC, one specific for California.
Hello.
Oops, sorry, Brian.
And then our new label that is registered everywhere in the US except California. We think at the end of the year, we'll have this new label that I'm going to be showing here in California. These are the crop groupings. We've done a great job of dividing out the crop groupings, the application methods, and any of the restrictions that we have. So it's a very broad, easy to use for your specific scenario. Some of the highlights are here. We have the boracic crop groupings, cucurbits, fruiting veg, leafy veg, our bulb vegetables. So for your onion and garlic crops, root and tubulars, specifically your sweet potatoes, potatoes, and carrots are all on this label. And then moving on to the next slide, we have a couple different application methods. So really this first one is application to field soil pre-plant. We're finding some new data where a treatment before you plant, very similar to your fumigant application timing, you could use LALNIX and it significantly reduces the egg populations because of our activity on the eggs itself. One of the caveats, if you are going to be doing a pre-plant application, is soil temperature. That I think is going to be our biggest restriction. If the soil is too cold or too hot, I think that could affect the activity of our purpose psyllium. So really reach out to your representatives. Let's tailor a specific program for your region based on your soil temperatures for that pre-plant application. Moving down on this list, we have an at plant. Oh, back to the other side, please.
Sorry.
Oh, sorry. We have our at plant application, typical germination water. It can be if it's a transplant, it could be a transplant dip. We have our application to soils post-plant, applications to orchards and vineyards, individual or existing orchards and vineyards. So some of your almond trees, your young almond trees, citrus, or grapevines, this product is labeled for that use. And then moving on to the next slide. I really wanted to highlight, we've broken up the crops, the insect or the nematodes. If you want to play the graphics here. So we have our nematodes separate, and then we have our crop groupings, and then also the application method all separate out. So specific crop groupings aren't restricted to specific application methods. It's a broad, encompassing label with minimal restrictions, making it ease of use for you in the field, is what I like to highlight. And moving on. The labeled chemigation. So chemigation is the majority of the way that this product is going to be used. So I really wanted to dig into the chemigation verbiage that we have on our label that allows you some different chemigation application methods. So first of all, we have this drip systems verbiage on our label, which encompass micro-irrigation systems. We also have a sprinkler systems where we specifically have solid set, center pivot, or impact or micro sprinklers on the chemigation section of the label now. And then for any guys that are doing the flood and furrow irrigations, we have our gravity flow systems that are written out on the label. So there's no gray areas, very cut and dry, allowing you guys to apply this product. And we recommend applying it halfway through an irrigation set. You want to chemigate it into pre-moistened soil, and it helps reduce or increase that soil temperature, depending on which growing region you are. And achieving that saturation into that root zone helps doing midway through the irrigation set. And then make sure to follow the LALNIX rehydration protocol, the video that we saw. The rehydration protocol is really going to improve that efficacy and performance of this nematicide. Moving on. Here's the states that it's registered in. All of the US except Hawaii. We are pending registration in Hawaii. And like I said, we have our old existing label registered in California. The new label is registered for the rest of the US. End of the year, we should have that new and improved label for California. And as soon as we get that California improvements, I will make sure you guys are aware of that. Right now, our label in California, it allows adequate use. I don't see too many restrictions on the existing California label. And then low toxicity option. This goes back to Megan and I's discussion about replacing some of your harsher synthetic materials. If you're looking to lower that toxic load, some of that worker safety application, it increases the speed and application there during planting or on your farm. LALNIX is a great option because of the low toxicity this product has. It's a signal word of warning. It has an REI of 24 hours. And here is the PPE that's required, a very short list. And you can see the picture. 0.4 millimeter thick gloves, eyewear, a respirator, and just long sleeve shirts and pants. So it's really minimal PPE, and there is no special training or certificates needed to apply LALNIX ACT DC. And next slide. Treatment timings. So we have our pre-plant before planting, 7 to 14 days would be the first window. Consult us on soil temperatures to determine if that's the right application timing. Our second application timing would be at planting through the germination water, or you could drench the trays or the transplants coming out of the nursery. And then our third application window would be during the crop life cycle of the product, 4 to 6 weeks intervals, depending on nematode pressure in your specific field. If you know that it has a history of nematodes, you might want to have a second application after that transplant water on there. And then moving on, just a quick little summary to wrap up this presentation. LALNIX ACT DC is an OMRI-listed bionematicide. It's based on the Perpetuum strain 251. It's the premium Perpetuum formulation on the marketplace, with the backbone being that strain 251, which is exclusive to Lymon. We are the only manufacturer with that specific strain and formulation selling into the marketplace. It's backed by 45 years of development. We've had continuous formulation improvements, field and lab trials, and we've really honed in the best use practices, such as this Lymon rehydration protocol. It's highly concentrated. You're not shipping massive amounts of product out to your farm. The use rate, 10.25 fluid ounces. So that's easy to applicate, easy to get out to that farm for your guys to apply. It's effective on a wide range of crops and nematode species, really targeting those eggs. We get some activity on juveniles and adult females, but that egg is really where you're going to want to target the application. And so if you're going to be doing an in-season application, you really want it to understand your soil temperatures and that life cycle of your specific nematode for that second application. So after those egg masses have hatched there in the root mass, you're going to want to apply as close to that second egg mass development in your crop life cycle. It's registered throughout the U.S., compatible with other microbes and beneficial nematodes, can be easily incorporated into an IPM program. And once again, that rehydration of those spores is critical with this product. And I think that wraps up our presentation for today. I think we've kept it right at, oh yeah, and here is our map of representatives. If you are not in California or outside of the state, please take note of who your Lymon representative is, and they can get in touch with them, and they can walk you through kind of on the soil temperature nuances, and also setting up trials out with your specific growers, treated versus untreated. We really want to show you the performance, real life, in your own fields and in your specific settings. So please do not hesitate to contact each one of us. And that wraps up. Everyone, thank you for your time. We hope that was informative and hope that the time well spent. And thank you guys again for joining us.